Today I wanted to talk about virus entry, and specifically as far as virus entry is concerned, what we're talking about is how virions attach to the cell, but not so much attaching to the cell. It's really about getting the genome into the cell. And lots of different viruses do this in different ways. And so this is really going to be, again, like the last couple of lectures, really an overview. We will start to talk about the individual viruses and how they do each of these things as we move along. But again, today is still pretty big picture. We will start with the individual viruses starting on Friday. So that's the overview cartoon-wise. In terms of just text, we have receptors. Receptor is, by definition, the molecule on the outside of the cell that the virion is going to bind to in order to get in. Many, particularly a lot of the animal viruses that we'll be talking about, are enveloped viruses. And so if you have a virus envelope, that's a lipid, basically a membrane around the outside of your virion, that then has to get through the host membranes. And it turns out that's actually a much easier thing to do than if you have a naked virion. And we'll talk more about that a little bit later on. A lot of this process, again, you have the binding of the virion to the receptor, but then you also will have the release of that genome from the virion. And that process has to happen as well. Often people talk about this as an uncoating process. Once the viral genome gets inside the cell, sometimes it can be replicated immediately there, but in many cases it has to get to the nucleus as well. And where you have that first interaction with the membrane is not going to be where the transcription, translation, and particularly DNA replication is taking place, which is all taking place in, we'll start out with the first one here, Corin Kieser, I think. Corin here? No? Okay. Priscilla? Herrera? I'm probably mispronouncing these names. Hillary? Is nobody here today? Diana, I know you're here. So where does transcription and DNA replication take place? Exactly. So you've got to get from the plasma membrane to the nucleus. So see, the easier questions. It's a really easy one. So they got to get to the nucleus. And we'll see, actually, it's even true for some of the RNA viruses that just have RNA genomes. And that's actually kind of bizarre. Why would you want to have an RNA virus that can actually serve as a messenger RNA? You shouldn't, in theory, have to get to the nucleus. But we'll talk more about some of those a little bit later on. And then towards the end of the lecture, I want to talk a little bit about some antiviral drugs. Many antiviral drugs are, in fact, directed at this step, the viral entry step of viral replication. And if these work, it's great because you never even get a viral infection in the first place. And we'll talk about some other viral drugs later on where you're having the drug acting at some later step in the viral replication process, which is a much less efficient one. So if you can actually get a viral drug that works really early on, that's a great way to do it. So this is the overview from our textbook in terms of how virions are being produced and then eventually get to the target cell. Again, remember we've got this extracellular space. This is the virion. The whole virus life cycle, of course, is the replication of the virion in the genome, etc. So very often you'll have, and these are for some of these enveloped viruses, you'll have the genome in a capsid that then gets an envelope added to it. And this is a nice example of where you get the envelope added in a different compartment rather than at the cell membrane. And this now envelope virus ends up being released on the outside. Quite a few viruses will have the viral capsid that just gets enveloped by budding at the cellular membrane. So you end up with a virion with an envelope on the outside of the cell that can either fuse directly with the membrane of the target cell, or it can get picked up by endocytosis. And this process, endocytosis, is probably the most frequent of any of these processes in terms of the virus is getting inside the cell. And we'll talk about why this is a useful thing a little bit later on. So there's a nice animation that I found here, actually from a different textbook, looking at the animation process, say the entry process. That's this one right here. I've turned off the sound, where you have an enveloped virion, we're going here, which will then interact with the receptors. Remember, that's what these are, extracellular molecules. These could be proteins, these could be lipids, these could be carbohydrates. And then you have fusion of the two membranes that takes place. The viral capsid is released. Then you have uncoating, which here is just the magic step of it disappearing. And then our viral genome is ready to go. You can also have, and again, this is probably the most common mechanism, you'll have binding to the receptor, and then this whole virion gets taken up inside the cell by endocytosis. And then once it's inside the vesicle, then you have membrane fusion that takes place. The virion is released, and then magically it becomes uncoated. So this is what happens with enveloped viruses. Again, either fusion at the membrane, let me stop this real quickly, back up a little bit, or you can have endocytosis and then release from those endosomal membranes later on. Naked viruses, and there are definitely some of these that have naked virions, they can't immediately fuse with the plasma membrane. There's no way that they can do that. So there are a number of different ways that they have to get through the plasma membrane. Again, probably the most common is this receptor-mediated endocytosis. And receptor-mediated endocytosis, by the way, these are all cellular processes. So cells have to interact with their environment. So receptor-mediated endocytosis can also be a, I don't know, food product, or if you're a macrophage, you're dealing with a cell that your bacterium, which you're going to then break down later. So these are all cellular processes as viruses have evolved to take advantage of some of these cellular processes. So here, if you have a naked virion, it can also then get inside the cell through receptor-mediated endocytosis. And again, here's an example where we have receptor-mediated endocytosis. But now you can't have membrane fusion that takes place. Somehow this naked virus, and again it dissolves here conveniently in this particular animation. We'll talk about how some of these things happen for some of these actual viruses a little bit later on. So there's the animation of how that works. Let's talk a little bit about some of the receptors. So the receptors, again, these are all cellular proteins that are involved in some other cellular process that has nothing to do with the virus and the virus infection process. Not surprisingly, all of these extracellular molecules that are used as viral receptors have some other kind of job. And so the case here for HIV-1, chemokine receptors. So CXCR5, CXCR4 for HIV. These are normal immune system signaling molecules. So chemokines would say, hey, there's an infection. You need to turn on the T cell for some reason. And that's what this protein normally does. CD4 is a classic case of something that's present on the outside of immune cells. How many of you had immunology? Not me. So you probably know more about this than I do. But these are specific recognition elements, again, that are present on the outsides of cells. And in many cases, these recognition elements have something called an Ig fold. And that's what this sort of omega here is supposed to represent. Ig for immunoglobulin. And immunoglobulins turn out to be really good ways of binding things. So it's not surprising you have an immunoglobulin fold on proteins in the outsides of cells, which are going to be interacting with other things on the outside of the cell. And again, these are things that viruses have evolved to deal with. For poliovirus, you have another example of one of these proteins that has Ig folds. This is, curiously enough, was originally discovered as being the poliovirus receptor. Of course, the cells aren't usually there to be infected by poliovirus. So much later on it was found what this normally does. CAR here, the Coxsackie and adenovirus receptor, again discovered as a receptor for a virus, but is much more important for other processes, again, mostly just cell signaling or getting things to come inside the cell that the cell actually wants to happen. Usually the virus is not the case. There are also a number of receptors that are not containing these Ig domains, integrins. The name here should sound very familiar. What does it do? It integrates things from the outside of the cell to come on the inside of the cell, and a whole number of different things. Another one I wanted to point out here, and we'll talk about these more later on. This particular one is an aminopeptidase, and so peptidases, what do? They chop up proteins, and so this one actually seems to chop up proteins in order to actually help the virus get inside the cell, but normally it's being used to break down peptides that the cell needs in terms of nutrition. So these are just, again, a number of these different kinds of receptors, and hopefully everyone saw in the news that New York is actually now requiring people to be vaccinated for measles. Very good idea, and we'll talk a little bit more about measles a little bit later on. So this is a ridiculously complicated slide, again, from our textbook, but I think this is great review. So when we talk about any particular virus, this is a great image to go back and think about in terms of virus entry. So we'll talk about some of these here as we go through it. Probably the classic example of an enveloped virus, here we have HIV, which has a nice capsid structure as well as an envelope. HIV fuses at the plasma membrane, releases this capsid. Because HIV is a retrovirus, it requires getting into the genome as double-stranded DNA in order to be able to replicate. So it also needs to be transported to the nucleus. And we'll talk about the nuclear transport mechanisms when we talk about HIV much later on in the course. And so here we have this replicating in the nucleus. Influenza virus is also an enveloped virus. This undergoes that receptor-mediated endocytosis process. Here again you have an envelope virus. That envelope ends up in the endosome. Then the virus membrane, the virus envelope, fuses with the membrane in the endosome. The virus genome, which is actually packaged with proteins as well, is released, and this then ends up going into the nucleus. And this is one of those examples of RNA viruses that ends up going into the nucleus. And we'll talk about why that is a little bit later on. Herpes virus is another one of the enveloped viruses which fuses at the plasma membrane. Hepatitis B virus is an enveloped virus that gets into the endosome. And then most of the naked viruses, particularly adenovirus and SV40, these are some of the naked virions that we'll talk about. Those also get inside the cell through receptor-mediated endocytosis. That last one of the animations that I showed. Those then have to somehow get out of the endosome. And one of the ways that they get out, which is in the case of adenovirus, is there are actually proteases which are in the endosome, which will chop apart the virion, and then there will be parts of the virion which can then help fuse and get out through the endosomal membrane. In the case of SV40, actually seems to be using not now a endosome, but other intracellular vesicle processes through the smooth ER, and those are vesicles that it just seems to transport itself in the vesicles and end up in the nucleus itself. Parvoviruses, small, naked virions, also get into the endosome and then release from the endosome. It's actually not entirely clear how those parvoviruses get out. But again, this is mostly just supposed to be a nice review of those different techniques, and then when we talk about each of these individual viruses, you can go back and look at this image in terms of thinking about the virus entry process. So we've talked about the entry process, we've talked a little bit about what those receptors are, what are the virus proteins that interact with those receptors, and probably the best understood of these, I don't know if I turn the lights down for this, maybe a little better. Yeah. This is probably the best studied of all of the viral receptor binding proteins. This is the so-called hemagglutinin, which should sound really familiar because what did we talk about hemagglutination for earlier on? Cameron? Cameron here anywhere? Caleb Price? Yeah. Exactly, right, for the hemagglutination assay. And so one of the great things you can use the hemagglutination assay for is for studying flu, and then looking at various different flu. So make sure I check off that you've actually answered questions here. Come back and find the rest of you. So HA is formed on influenza virus virions as what's called HA0. And I'm sorry about the nomenclature here again. It's all the different virologists who've come up with them in the first place. The actual form that is present on a mature and infectious virion is actually a combination of HA1 and HA2. And so HA1 and HA2 are labeled here as being blue and red. HA0 is just a protein fusion between these two, and there's a proteolysis site right here in HA0, which then will give you the separate red and blue proteins HA1 and HA2. HA2 is this piece at the top here. This binds to the receptor on the outside of cells, and certainly for hemagglutination it's present on the outside of red blood cells, but these are normally infecting epithelial cells. Turns out that what it binds to is sialic acid. It's a carbohydrate that's present on the outside of many different cells. What happens is once it has that binding, that will help bring it inside the cell. This goes in through receptor-mediated endocytosis. But then once it gets into the endosome, endosomes acidify. And this is one of the ways that endosomes normally happens in cell biology. It's to help break down whatever's come inside the endosome, usually going to be food. But when you have that change in pH, the virus recognizes that change in pH by causing a change in the structure. Let's go back to this. And that change in structure here, it's a little hard to see, but you have a protein, part of the HA1 protein here, which then isomerizes and basically sticks a hydrophobic piece out at the end here. And this hydrophobic piece will stick into the host membrane and then allow the viral membrane and the host membranes to fuse with each other. Yeah?
Is this the same thing that's happening when you have fusion on the plasma membrane from like an HIV? It's like the same kind of like opening mechanism, but is it the same type of receptor?
Yeah, so I'll try and paraphrase your question here. And so there are two things that are going on here. We have a receptor, and that's what's the cellular protein, in this case actually sugar, that the virus receptor binding proteins can interact with. And these virus receptor binding proteins, again, that's what they do is they bind. Sometimes you have a separate fusion protein, and that's what this is serving as here, or you can often have the two of them together, like hemagglutination. So this binds up here and then has a fusion part here. And so this has a fusion and receptor binding protein in one. In other viruses, you actually have separate proteins that will do those jobs. So we're really talking about the virus end of things. And we're just about to look at some animations which will hopefully clear that up a little bit. And that hairpin mechanism, again, how you're getting the membrane fusion to take place, really has to do with first these kinds of conformational changes, the conformational changes that will happen in these fusion proteins, but then further conformational changes that will pull the two membranes together. And that's what we're going to take a look at here in these next animations. So we'll take first look at this one, which is the HIV fusion mechanism, again happening at the plasma membrane. So here, now this is the fusion protein in HIV. It's the viral membrane is down here. The host membrane is up here. You have a conformational change which has happened, again, due to receptor binding. This is just one of the three of these. And then these fusion peptides. The fusion peptides, these are those hydrophobic things that will stick into membranes. Usually they won't stick into membranes until you have the receptor interactions as well. Again, could be the same protein, could be a different protein. In the case of HIV, it's actually a different protein. And what happens is first you have the fusion peptide stick in, and then you have a conformational change. And so this is the conformational change, which is bringing those two membranes together. So here, nice animation thereof. You have the fusion protein, which has interacted, and then you start to have this conformational change where these alpha helices are forming and thereby pulling the two membranes together in order to get membrane fusion. And this is that, so we think you're talking about the hairpin for getting these things to come together. First you have one of the leaflets which is bound, and then both of these, which then will allow the release of, in the case of HIV, this would be the capsid, which is present on the inside. In other cases, we'll see it's actually the genome which gets released. These all come apart, and then, of course, the virus capsid can come through here. This is also a nice example of how people are trying to develop drugs to deal with this kind of process. If you know exactly molecularly how all of these things are moving around, maybe you could design a drug that will stop these kinds of changes in the proteins. And so this is an example of one of those drugs, and basically what it does is it blocks this conformational change, so you can't get the membranes to come together. You can't get membrane fusion to take place. I don't think this one's actually in the clinic, but an idea of trying to use what we know about structure to design some antiviral drugs. Again, same kind of thing here. This one I think works actually much better. Peptides are not great drugs, but these small molecules end up working out pretty well, and don't think this is, again, currently being used, but I know it's one of the ones which is under development in terms of anti-HIV drugs. So that's the case for HIV. We also have an example now of dengue virus. Also binding to, now this is going to be in an endosome, so it's a pH-mediated change where you end up getting membrane fusion that takes place. And instead of a capsid being released, it's going to be the individual genomes which are getting released here. And I do need to go ahead a little bit with this because it talks all about how dengue is a nasty disease, which we'll hear all about later in the course, but basically about here where we can start to see the fusion taking place for this. I'm actually working with a guy at OHSU to try and make vaccines for this. So that's the dengue virion, receptor-mediated endocytosis, gets into the endosome. Those endosomes are transported inside microtubules, inside the cell. But what's happening is now you're getting acidification that takes place. And so that's what these bits are supposed to be here. Really big conformational change that happens in the surface protein, also known as the envelope protein, in these viruses. These trimeric forms are actually now extremely similar to that trimeric form that you have in HIV. Then you have a very similar kind of conformational change. You see these alpha helices forming, pulling the two membranes together. And once they've pulled the two membranes together, then you have the release of the virus genome, which is actually animated in this case. And you can see all of the happy virus genomes coming outside of the cell. Here they are, coming outside of the, outside of the endosome. And these can be translated immediately, and you end up replicating more of these flaviviruses. So all of these are linked appropriately. Yeah, Christian. So the question is, are they using snares in terms of what's known about vesicle transports that you have in cellular vesicle transport? You probably know more about snaps and snares than I do. There are some viruses that do use those, but these particular ones do not. Yeah?
So on the HIV video, it said it usually only requires one of the trimer to actually create that fusion. Is that normal? Or you don't need all of them to make it happen?
Yeah, so I think the short answer to your question is I don't know. The long answer is I'm not sure that anybody knows, and actually analyzing exactly how many of these you need to get fusion to take place is a great question. That animation that I showed of HIV is actually from Stephen Harrison's lab, who's a professor at Harvard. And they're doing a lot of research into trying to figure out exactly how many of those molecules you need to actually get membrane fusion to take place. We do know in the case of dengue that you need the trimeric form in order to get fusion, but whether you need 2 trimers, do you need 4 trimers, do you need 8 trimers, I think that's really an open question. I think for HIV they know a little bit more about it, but I think it's not just the 1 trimer. I think you do need multiple ones. Yeah, in the back.
In terms of HIV drugs, like you said, is that how post-exposure prophylaxis works?
Okay, we'll talk more about post-exposure prophylaxis and just sort of general anti-HIV drugs later. What I'm not sure is if they're using these entry drugs as part of the cocktail. So it's actually multiple different drugs that are being used to try and address this. I don't think that they're fusion inhibitors, but some of the newer, they keep changing actually the composition of what they're using. So the ones that I know about are integrase inhibitors, reverse transcriptase inhibitors, and protease inhibitors. I know that those are being used. They may be trying to use some of these entry inhibitors as well. That I don't know. Okay, more questions for me? I can ask you one now. See if we can get that part of the technology to work. Let me get that piece out. So let's move to the next slide, if it will. No? Okay, now we're not talking to each other. Fine. Okay, we'll do our clicker question then. Here's a little bit of extra time while I get my clicker thing out here. Fusion of animal virus envelopes with host membranes during infection mostly happens due to which of the following? Proteolysis of fusion proteins, structural changes in fusion proteins, pH-induced aggregation of fusion proteins. I can just start this over here, be easier. Yes, it's AA. Close. Structural changes in fusion proteins, pH-induced aggregation, nuclear localization of fusion proteins, endocytosis of fusion proteins. As usual, if you don't get above 70–80%, we'll let people think again and discuss with their neighbors what they think. And as reading the question, I think there may be some confusion about it here as well. So. Okay, not quite consensus, so why don't we stop and quickly discuss with your neighbors. Only 30 seconds should be enough. Decide what you want to vote for. Okay, we ready to vote again? Okay, our marks get set, go. Again, you can continue to discuss this time. No quiet required. I'm always hoping for the 100%. And usually it seems there's at least one person who's trying to stop it being 100%. Which I will just try and consider that it's someone who's just really out to get me, not that anyone doesn't. They probably actually know the answer and are just trying to spite me. So 24 people here today, more or less, or 24 people have clicker questions or clickers with them. Okay, so close. So close. Yes, it's the structural change. And so the key to this question is we're talking about fusion. So the fusion of the membranes is going to be those conformational change that happens with those fusion proteins. So B is correct. That reminds me, I have all of the, just a second. I have all of the clickers, I have all of your points, but somehow the clickers and your names haven't gotten hooked up to each other because the link that I put in D2L doesn't seem to be working properly. I will keep working on that. But we've got all the clicker ones. Yeah?
Yeah, just curious, you said, I think might be some confusion with the question.
Yeah.
What did you see?
Where I see so it's due to which of the following. And so I suppose one could argue, and probably whoever the two people were who picked this last one here, is that the endocytosis then would be that process, and that you'd have to have that. But since not all viruses like HIV, some of them are fusing at the plasma membrane, so you wouldn't have endocytosis that was happening there. So that was the confusion I see. When I go back and look at these questions, I'm like, oh, they could have written it this way, could have written it that way. And as I've mentioned before, for those of you who are were in molecular biology with me, the exam questions get much better proofread than the clicker questions. So hopefully there's less issues then with that other than what we have here. So got this working again too. It's also a nice thing about clickers. I have some time to play with my technology, try and get it to work. So as I mentioned, there are a number of virions. They've released their genome inside the cell, but that's not where it's going to replicate. And so why do you need to get to the nucleus? As Diana pointed out, that's where you have replication going on. So if you've got a DNA virus, it makes perfect sense that that DNA genome has to get to the nucleus. If it's a retrovirus, you have a DNA intermediate, that's got to get to the nucleus. But why have an RNA virus, particularly, it doesn't actually really matter, positive strand or negative strand virus, why would you want to have one of those go to the nucleus? Because it can replicate itself. It can translate as a positive strand RNA virus. By definition, that's something which can be translated. So it turns out that most of the RNA synthetic machinery, again, most transcription is taking place in the nucleus. So all of the precursors, all of your ribonucleotide triphosphates are in the nucleus. And it turns out that there are some things that some of the viruses require. We'll spend a lot more time talking about this when we talk about flu later. Need some other parts of the machinery which are present in the nucleus and not present in the cytoplasm. So some RNA viruses have to get to the nucleus, but some RNA viruses replicate perfectly happily in the cytoplasm, and there are even some DNA viruses that replicate perfectly happily in the cytoplasm. And I'll let you think about how that can happen a little bit later on. So how do they get to the nucleus? There actually are a number of different mechanisms for how you can get your either genome, or in some cases actually even the whole virion, into the nucleus. Nucleus, of course, has a membrane around the outside and a nuclear pore. And what are nuclear pores do? So we've got someone next on the list. Jonathan Wynn? Jonathan here? No? David, Ross, yep, pardon? Yeah, sorry, multiple Davids, that's confusing now. So what's the nuclear pore for?
Regulates which molecules get in.
And is usually a viral genome or a viral capsid something you want to get in? No, exactly. So viruses have figured out to totally over-anthropomorphize here, evolutionarily, different ways of getting inside the nucleus. And one of them is actually just to hang out. Wait till the nuclear membrane dissolves. That's a way of dealing with the nuclear pore. It's gone. And so then when the nucleus reforms, you're inside the nucleus. And so that works. Turns out that some lentiviruses, not HIV, interestingly enough, but other retroviruses actually will wait for mitosis to take place, and then they can get inside the nucleus that way. Some have some very specific proteins that will help them get into the nucleus, and so nuclear transport. Your nuclear pores, you've got nuclear importance, nuclear exportance. So if you've got an importance signal, a nuclear localization signal on your virion or on some part of it, that will get that genome into the nucleus. Some cases, adenovirus is a good one. The capsid is transported to the nuclear pore, but it's too big to get through, and so that metastable state of the capsid then degrades, and then the genome is released at that point. And in the case of some really small DNA viruses, the whole capsid can actually get through the nuclear pore complex. And so then on the capsid, they've got nuclear localization signals. And in fact, the very first nuclear localization signal ever to be characterized was a viral protein for getting it into the nucleus. So that's the process for getting into the nucleus. What we haven't talked about yet is how to get into bacteria. We talked a little bit about it last time with this syringe process. Getting into animal cells usually is relatively straightforward. Membrane fusion works really well. And that's usually because animals have other ways of protecting themselves because they're multicellular, and actually a lot of these viruses will just get into specific cell types. On the other hand, bacteria, and we'll talk more about plants as well, talked about it a little bit last time, they have a really tough outside because they've got to survive by themselves in the big bad world. And so the virions somehow have to get their genome into that. And so if you've got a gram-positive, it's got a really thick cell wall and one membrane. Gram-negatives have two membranes, the outer membrane and the inner membrane. So somehow you've got to get through that. And my favorite example of this is the bacteriophage T4. A bunch of you have probably seen this before, but it's an absolutely amazing, as far as I'm concerned, animation looking at getting T4 through a bacterial membrane. Here are the poor bacteria, the E. coli here in the background. Here's the bacteriophage T4 at a almost atomic resolution. Each of these individual proteins, they know very well what all those structures are. And so here comes your T4 floating around. Eventually we'll encounter an E. coli with its long tail fibers. That's what all of these guys are here, the long tail fibers. Those interact with receptors on the outside of the cell. There's a conformational change that happens there. These short tail fibers then interact. Then you have yet another conformational change where literally it drills through the membrane and drills through the outer membrane. Then there are proteins that will break down the inner membrane. Then this tube, which allows the rest of the genome to come in. So I'm going to see if we can watch this again. We can go through the process. So again, there's a lot of similarities here. Conformational changes that are happening and interactions with receptors. Didn't mention this so much, but you may remember when we looked at those different receptors for HIV, there are two different receptors, CD4 and the chemokine receptors. Here also multiple receptors are being used. You have the short tail fibers and the long tail fibers, and then this really amazing conformational change that goes through here. And there are some enzymes. That's what these green ones here, enzymes that come in order to allow the rest of the genome to get inside the cell. I first saw this animation, postdoc who was in Michael Rossman's lab at the time. We're at a meeting in Florida. Just a second, Chris, I'll be right with you. And he showed this video, and everyone's like, "Oh, wow, this is amazing!" And it came out in Cell a couple of weeks later. And then afterwards at the bar, when I was having a beer with him, he said, "Actually, there's a problem with the video." Anybody know what the problem with the video is? They heard me talk about this before. So when they did the animations, they got the clockwise versus anti-clockwise in terms of how it compressed to go through the membrane wrong. And the researchers told the animators, said, "We're not changing that. It's not happening. You're not going back and changing that," because it would have been way too much effort to make that change. Yeah, Christian. Mm-hmm. Okay, so the question here has to do with basically how that conformational change is taking place. And is there some kind of nucleotide hydrolysis that's required to have that happen? So that's a really fabulous question. And if you think about it, this is all happening extracellularly, or even if it's inside the endosome, it'll be happening inside the endosome. Is extracellularly, are there any nucleotide triphosphates floating around? Energy-wise? No, so there are not going to be any. And so this gets back to what Nacho was talking about metastability. And so there's basically energy which is stored through the assembly process, and you kind of think of this like a compressed spring or a cocked gun or something like that, which is ready to change, but is only going to change when you have a particular kind of interaction. So there's no nucleotide hydrolysis that's happening as you have these conformational changes, but they're basically set up to have these conformational changes. And so it's kind of a bizarre way that at least everything I always think about in terms of protein structures, that you got this nice stable protein structure, it doesn't change. Maybe if you've got some kind of hydrolysis, big change that happens, but no, this is in a kind of a constrained state, which can then change really easily. And as you see for some of the animations, you have a really big conformational change that can happen. But it's a you're loaded or metastable state thinking about virions. So thanks for asking that question. Okay, so now I wanted to finish up chatting a little bit about some antiviral therapies. We saw the one already in terms of that conformational change in terms of HIV and the HIV fusion protein. There are many different ways, and actually probably the best way to protect against viral disease at all is to have a way to block the binding in the first place. So no interaction with receptor. If you have no interaction with receptor, you're never going to have any fusion proteins, none of the rest of it. So that's the best way to do this. And the best way to have that happen is you have your antibody response or your T-cell response that already has been juiced by vaccines. So vaccinate, vaccinate, vaccinate. But that's literally how vaccines work. Vaccines work by blocking the virion before it has a chance to interact with receptors. So you can say, vaccines are really blocking that entry step. And as I talked about a few minutes ago, if you can block the entry step, all the bets are off. So it's a great way to do it. There are some people who are trying to use the same kind of mechanisms that you would get with a vaccine, but now instead have a virus receptor binding protein antibody, and so specifically design an antibody, make antibodies, and the technology is getting pretty good. You can make monoclonal antibodies. There are quite a few therapeutics now with monoclonal antibodies. And so trying to make antibodies that will block that interaction happening. You can also try and swamp the system. And so this is actually one of the anti-HIV mechanisms that people are trying to use, is use huge amounts of the receptor just by itself, but not associated with the cell. So basically mop up or use a sponge to grab all the virus particles before they have a chance to actually find a cell. Unfortunately, this process hasn't worked very well, and a lot of it has to do with the fact that making big proteins like CD4 or CXCR5, which are the HIV receptors, is really hard to do and actually get them to where you need to have them. So vaccines, great. If you can get some way of blocking the interaction, that's wonderful. You can also have drugs, and some of these drugs have worked quite well, block the uncoating step. And when I say the uncoating step, that's where in that very first animation where the capsid sort of dissolves, and so you have the release of the genome in some way. In the case, and we'll look at some examples of these in just a second, for flu, what's happening there is flu has this conformational change once it gets inside the endosome. And what's happening inside the endosome? Who's next on my list? Daniel Ziegler. What happens inside the endosome? Causes the conformational change? So, yeah, the proteins are going to interact. That's getting the virion inside the endosome in the first place. But what happens in the endosome that causes the extra conformational change that gets the two membranes to fuse? Yeah, it's the change in pH. And so it turns out that that change in pH is what happens with pH. And what's a change in pH? What happens then? What's pH all about? Yeah, denaturing the proteins. But literally, it's the concentration of hydrogen ions, right? And so it's literally, you're looking at hydrogen ion transport. So it turns out that these virions actually have channels in them for transporting hydrogen ions. And so that's how they're sensing that there's a change in the pH. And it turns out that the one in flu is something called M2. And we know the crystal structure of M2, and we can design inhibitors for that. Amantadine actually used to be a really good anti-influenza drug. Unfortunately, almost all of the circulating influenza strains now are resistant to this because it was used so much. Rhinoviruses. Rhinoviruses are common cold. These have to uncoat in a conformational change of the capsid. These are naked, unenveloped particles, and in order to get their genome to release, they have to have a pretty big conformational change. And this particular protein is involved in blocking that conformational change. Same thing is true with some other picoRNA viruses. So let's look at some of these structures. This, as I mentioned, is the M2 protein. It's a pore. This is now, normally it wouldn't have this small molecule in the middle of it here. This is where those hydrogen ions are going to be going through to get the acidification, actually the interior of the virion. And this small molecule here was designed to be able to block that and did really well until the virus mutated, and then these particular amino acid side chains are different, so it doesn't bind to this drug anymore. This is now one of those small RNA viruses. There's to be a big conformational change, and we'll see this actually when we talk about these small RNA viruses a little bit later on. When you have an interaction of picoRNA viruses, any of these rhinoviruses with the outer membrane of the cell, they actually undergo a conformational change, not unlike what you see with bacteriophages. And that conformational change then allows a pore, makes a hole in the membrane that the genome can come out through. And the way that that conformational change works is through this, change in this protein right here, this dark blue one. And if you put in an extra drug, which is in pink here, that cannot move anymore. That can't undergo this conformational change. You can't release the genome inside the cell. So that's a potential anti-poliovirus medication. So there are some other mechanisms which can be used. We'd already talked about, Daniel mentioned the acidification which happens in the endosome. You can actually block endosome acidification. And there are drugs that you can use for this. These, however, end up having some big problems, and that is you need endosome acidification for the normal function of an endosome. And if you block endosome acidification, then you're going to have all kinds of other problems that are happening relative to the rest of the cell. There are other, and we'll talk more about the activities of this later on. Tamiflu is one of the few anti-influenza drugs, although people are developing more, that actually works. This is a inhibitor of one of those enzymes. And so we saw the enzyme in the case of bacteriophage T4 breaking through the membrane. Here, the neuraminidase, neuraminidase is a enzyme that's on the outside, actually in the envelope of flu virions. What it does is actually chops off the receptor, so chops off the sialic acid. So why would chopping off a receptor be useful for a virus? Anybody's guess? I'm not going to put anybody on the spot on this one because it's a tough one to answer. I had trouble figuring it out. So why would you want to chop off a receptor? It's when you're trying to get out. So when the virions are trying to get outside of the cell, you don't want to bind back to the cell that you just infected. You want to go and find a new one. And so you can actually, what will happen is those virions will actually get stuck on the cell that they originally infected. And what the neuraminidase does is it actually chops off the sialic acid so that this virion can go and find another cell in order to infect. And this is the structure now of that particular enzyme. It's the neuraminidase with Tamiflu, oseltamivir, associated with it right here. And this is also exactly where the active site is here, where you would have that sialic acid cleavage event. So, more questions on this stuff? We ready for another clicker question? But hopefully I can get to work a little bit better this time. Okay. Most common cellular mechanism used for animal virion entry into host cells is which of the following? Phagocytosis, pinocytosis, endocytosis, hemagglutination, pore formation. We're still aiming for that 100%, right? We will get 100%. By the end of the term, we'll get 100%. I guarantee it. And I can sell you a suit. No. Five. Okay, we're still not quite at 100%. And again, I hope it's not somebody trying to spite me. Oops, where were they? Yes, but C is what I'm looking for, endocytosis. This is also happening with enveloped and non-enveloped viruses. There are examples of every single one of the other ones, but the most common one does seem to be endocytosis and how it's happening. Although hemagglutination, that's not going to be causing actual virus infection. As you mentioned, it actually is much more of a detection mechanism that's used for looking for virus infections. Yeah?
Hey, we got the 100%.
Oh, woohoo! Awesome, because I forgot to stop it. So, good for us. So, oh, no. You're messing with me. It's only Wednesday. Come on. Okay, can I stop it now? Stop, stop, stop. See? Still trying to figure out the technology. You would think after, what, 17, 18 years teaching, I would be better at this, but I guess not. Haven't been using the same technology all the time, though. So, didn't I do this in molecular last term as well?
There was one question.
Yeah, I think there was one last term where I did that too. So, okay, so did want to finish up here now. Actually, not more entry anymore, but talking about exit, getting outside the cell, and that was kind of the transition that we had with the neuraminidase, because neuraminidase is really important for getting the virions outside of the cell. One way that you can get outside of the cell is, and these are actually names that my graduate student came up with, now crash out of the cell. That's literally breaking open the cell completely and releasing virions from the inside. This is particularly important for the naked viruses, the ones that do not have envelopes, because if you're escaping from an animal cell, usually it's going to be a budding process. Well, if you're budding and you don't want to have an envelope around them, how do you have to somehow escape from that bud in the first place? And so very often the non-enveloped viruses can, or let's say not can, but do in fact break open cells. How does this happen? Usually it's a specific protein which is made as one of the late proteins, which is a lysis protein. And it will then cause cell lysis to take place. And we'll talk about exactly how each of those proteins work much later on in the course. Now, the key to this process is you've got to make sure that you make this lysis protein only at the right time. Because if you are assembling your virions and they aren't completely assembled yet, and the cell lyses, then you don't have any active virions. So it has to happen at an appropriate time. And so the timing is really, really critical. And we'll talk about this when we talk about those viruses that have lytic pathways a lot more later on, because this is a very important point. And how you can actually time expression of various different proteins and genes is actually really interesting. An easier way to go about doing that is also what he calls exfiltration, budding process for enveloped viruses. This makes perfect sense. And then the budding process is also something that cells are doing all the time. You know, they're releasing vesicles. The key to that is actually sort of the inverse of what you were talking about before, the hairpin structure that forms in terms of getting the fusion to take place, where you also have to have this budding process where you seal off the necks of each of these particles as they are being released. And so that's a really interesting question on how that works. But your timing's not critical. You make a virion, it gets to the membrane, it buds off, it's good to go, and you can continue in this process. However, you have to make sure that you have all of the virus envelope proteins in the right place. If you just bud willy-nilly off of the surface of a cell, then often you're not going to have the envelope proteins. And what are those envelope proteins really important for? And who are we at next? Crystal? Yeah, so what are those viral envelope proteins mostly going to be important for? Yeah, so they'll be important for protection. Usually it's the envelope which is mostly important for protection. So what are those proteins going to be doing? Like the HA protein, for instance? Yeah, well, they're the receptors, the receptor binding proteins. And so if you don't have a receptor binding protein, how useful is your virion going to be? Not very useful at all. So making sure that all of your virus envelope proteins are in the right place, this is what I call the 2D assembly process. And so in the membrane, you've got to make sure that your specific viral envelope proteins are in the right place before you bud in that particular way. Because if you budded wherever on the surface of the cell, that would be a problem unless you've completely covered the cell in your envelope proteins, which is kind of hard to do. You'd have to make really, really large amounts of them. One thing that we'll talk about when we talk about the archaeoviruses right at the very end of the course are these really amazing things called VAPs. What are they? I'm trying to remember what the abbreviation is for now. Virion-associated pyramids. And so what happens with these, so another way of getting outside the cell is literally to make a hole in the cell. And the way that these particular virions do that is they'll assemble a 7-sided pyramid that pokes through the membrane that they will then open up and make a hole that the virion can come out of. Absolutely incredible process. Yeah. Okay. Pardon? Oh, pyramid. So it's a 7-sided pyramid. And again, we'll talk more about that when we talk about those archaeoviruses very, very much at the end of the course. It's a sort of brand new exit strategy way of getting outside of the cell. So thank you for receptor binding proteins. So how you get recognition, it's that receptor binding protein on the outside of the envelope. Or you can also have all the naked virions have exactly the same issue. They still got to find the right cell. So there'll be some kind of receptor binding protein that's present on the outside of the virion. How do you get in? Either through a cellular process or a very specific viral process. So again, receptor-mediated cytosis is a classic cellular process, but making holes in the outside of a cell is usually not a cellular process. So that's the different way of doing it. Uncoating, uncoating here is basically a genome release. This can be some kind of membrane fusion event, or you have a metastable state of your virion, which then releases the genome. After that, nuclear entry, again, this can be dissociation of the nucleus or tricking the nuclear pore complex in terms of getting in, and then there'll be a number of drugs, and particularly also vaccines, which will block this whole process so that you can get the virion entering into the cell. So with that, I think we're done. I actually finished all the slides today. What a concept. Cool. So yeah, we will start talking about individual virions and viruses starting on Friday. See you then.